All right, everybody, we're live. We got a ton of stuff to cover. Are there any questions about anything? All right, let's get moving. So the last thing that we did in the last class was the Punnett square for the Labrador Retrievers. The correct answer for the Punnett square is this one. Record that display. Is this. This is how it should have been. They were dihybrids, which means they were heterozygous for both of the traits, so they could have made either of any of these four contributions to the offspring, right? So you line those up. Your cell has your Punnett square has 16 cells. It's very similar to the independent assortment one we did. The only main difference is that there are only 3 outcomes for this because of the regulatory gene. You can only have black, brown, and yellow instead of 4 outcomes for independent assortment. Remember the P's, the green, smooth, yellow, wrinkled. So basically remember that in the example of the Labradors, if you had homozygous recessive for the second gene, which we indicated with ease, that blocked the pigment and produced a yellow individual no matter what the other gene was doing. Okay? If it's not homozygous recessive, then the pigment is allowed. Okay? So you get 4 blocks. 4 of them are blocked, so you have 4 yellows, 4 yellow labs. Now, the other gene determines what color it is, provided it's not blocked, and black was dominant. I gave you that information. So in order for it to be brown, it would have to be double recessive in this first gene. Okay? And if it's double recessive for both, the pigment's blocked, and you end up with yellow. So actually, yellow is more common than brown. Brown is the least common, and the ratio is 9:3:4. Okay? That was the correct answer. I realize I didn't give you perhaps quite enough time for it, so everybody will get partial credit if you got anything correct. This is supposed to be you. Now, explain that again. We are different how? We don't get it. Right? It's sort of supposed to be like a feel-good message about racial equality, if you didn't get that. Slightly more popular is this one with yellow lab, black lab, chocolate lab, and meth lab. Thought we'd end on a humorous note. Yes. The parents, yes. The answer to the question as to what color the dihybrid parents are is black, because if they're dihybrids, they're heterozygous for the block trait, which means it's not blocked, and black's dominant, so they were both black. If you got that right, you got a point or 2. And you got a point for the dog. And you get a couple more points if your Punnett square had 16 cells, and then you get 8 points total if you do all of that. Anne told me that it was a 9:3:4 ratio. Okay. I mean, I know I just terrified you with stories of how difficult the test is, but I will tell you there's no problem as difficult as this on the test. The only Punnett scores you do on the test are actually 4-celled, so they're easier. Yeah, that's why this is extra credit. So don't get too freaked out about that. I mean, you'll look like this when the test is over. don't get me wrong. All right. Let's see here. Now, let's get back to Mendelian genetics. This is where we last left off with our Labrador Retriever. Yes, that's the one I want. This one is this one. All right. We left off here. Okay. So we were talking about, this was an example of deviations from Mendelian inheritance. And we've been, bless you, going over a list of deviations from Mendelian inheritance. Okay? Let's talk about a couple more that are very important as well, besides regulatory genes, which are extremely important in and of themselves. One that's quite important that you probably have some familiarity with are sex-linked traits, kids. Sex-linked traits are things that are found on the X chromosome, not the Y. Okay, this is easy to confuse because males will express a sex-linked trait because they only have one copy of the X, which means that even if it's a recessive trait, it gets expressed in the phenotype for males because there's no dominant to mask it. And that's why females tend to be carriers for things like baldness, color blindness, and hemophilia, whereas males have the trait, right? Hemophilia is an inability to produce a protein that causes blood clotting. So if you suffer from an internal injury, you can internally bleed to death. Yes?
Why males are more likely
Because the gene for color blindness is located on the X chromosome, and you only have one copy, so it shows up in your phenotype. And in fact, we can give you a little color blindness test right now, although I want to assure you, I just want to be clear here, since I had a very bad experience with a student flipping out and losing their mind in this class. This is a color blindness test for red-green color blindness, which affects about 10% of the male population of the United States, or over 14 million people. And I need to say that I'm not a doctor. Well, I am a doctor, but I'm a doctor of anthropology, which means I'm not qualified to actually administer these tests. So do not think this is actual medical diagnosis. Because one semester I had a kid, and I was like, oh, this is a color blindness test. And, you know, if you see a certain number in this field, that means you're probably colorblind. And he just, like, wigged out, lost it. Like, he was just like, oh, my God, I'm going blind. Oh, my God. I was just like, no, you're not going blind. You have color blindness, right? That took a while to convey, but it was crazy. It was crazy. So I'm not a doctor. If you do not pass this test, you're one among millions, and it's okay. If you do not, if you get the result of this test that might suggest that you are red-green colorblind, I urge you to go onto the web and do mini tests, which is what they do when they really test you, and get tested by a professional. But the question is, what number do you see in the field, in the image on the left of the screen? Wait, it is a 5? Nice. The 5 and the S are the same answer. Don't get smart with me here. It looks like a fish. Yes, exactly. I see a man and a woman making love. All right. If you see a 2 in the pattern, it means you're probably red-green colorblind, because that means you can't see the 5, so you're not differentiating the blues and the greens in the field. It's okay if you don't see the 2. The problem is if you do see the 2, and that's when you should get online and do some tests. Yes. So anyway, I could spend more time doing more of these types of tests, but we don't have enough time, so we're just going to move on. The point here is that the information that determines this trait is contained in the portion of the X chromosome that is not present in the Y. The X chromosome has about 1,100 genes. The Y has less than 50. The Y chromosome, which is the thing that makes you a male, although it's not responsible for your genitalia, that's another story. In any case, the thing that makes you male, which is the Y chromosome you have to inherit from your father, your mother can't contribute one to you, is a little teeny chromosome that's actually kind of pathetic, actually. It doesn't do very much, and it's very, very tiny. It's actually much tinier proportionally to the X than this stylized diagram suggests. Now, I'll tell you something very interesting that links up with our film on Y-sex. Remember that Jeffrey Miller in the Y-sex film made the observation that the human brain may be a kind of peacock's tail, right? In other words, it may be something that indicates something about the genetic viability of your potential mate in an even more effective way than the peacock's tail shows you that the peacock is healthy. When you talk to a person, if they have a brain problem, it tells you realize this instantly and then perhaps cross this individual off the list of future mates. I don't know. It really depends what you're into, but that's cool. Okay? So the brain is a way that we get to know each other, right? And we assess whether or not someone represents a potential mate. Okay? Now what's interesting and what emerges as we study these two chromosomes very closely is that there is a very large number of brain-related genes on the X, and lots of various learning disabilities as well as mental health issues have genetic bases in the 1,100 genes that are on chromosome X but not on chromosome Y, which means that males will display these problems like a faulty peacock's tail, and the whole thing makes potential poor male mates more transparent and more obvious to their female partners. It also explains, according to some researchers, why, when you look at the distribution of IQ scores across human populations, you end up with a pattern where the average person, whether male or female, has an IQ of approximately 100. It varies slightly by different populations. But if you look at the distribution of female IQ scores, it goes like this, where 100 is also the peak of the number of people, number of women. Whereas the male, who also has males as a group, who also have an IQ average of 100, actually have a distribution that's more like this, with 100 being in the middle. Okay, I'm exaggerating it to make the point, but the point is that males have peaks at the extremes, but not in the middle, and that's partially as a result of the fact that these genes are expressed in the phenotype due to their location on the X chromosome. How's everybody doing?
Men are generally either really unintelligent or highly intelligent.
Yeah, they rank at both ends of the spectrum, either low or high, but not in the middle as much. It all averages out the same, but this is a result of the fact that all those problems get expressed in the phenotype.
I don't understand why. So women tend to carry them, but men express them all because of the Y?
Yes, because you have, let's say we're talking about a gene for, I don't know, color blindness. Or, actually, in the next slide, shows you the inheritance pattern of the gene for hemophilia in royal families, okay? So,
They went out, not down, right?
well, let me explain and then we'll answer your question. Okay, so this shows the inheritance of hemophilia in Europe's royal families, which are all intermarried with one another. Female carriers are half circles. Those who have hemophilia are shown in dark green. Okay, incidentally, a protein has been synthesized, so now if you have hemophilia, it's treatable. It has not been historically. And basically, even a small accident causes you to bleed internally until you died. So basically, you're looking at a disorder that's genetically based that would basically end the chances of the organism that suffers it, the person, from reproducing. Okay, the most famous case of this is actually the little Tsarevich, Alexei, who was the son of the last Tsar of Russia, Nicholas. And he had hemophilia that was diagnosed when he was a child. And in order to make sure that he did not die, his parents would not let him play, and they had him carried around by an attendant. Like he was literally carried by a person for much of his life. And that was because if he had suffered any kind of accident, he would have died because his blood wouldn't have clotted internally and he would have bled to death. Now, the whole thing was rendered moot when the Russian Revolution happened and they were all gunned down by assassins, which, you know, if you're gunned down by a line of assassins, it doesn't really matter whether or not you have hemophilia. You're going to bleed to death, okay? Just, you know, trust me on that one. So he made it to the assassination, but then he was killed. Now, the Russian royal family, like the other royal families of Europe, intermarried with each other, which is, I think, what you were talking about. So it was passed between the families, right? The whole thing apparently originates actually in a mutation that Queen Victoria had. It is not thought that she inherited it from anyone. It's thought that it was a mutation in her own genome when she was born, which she, as a carrier, had no idea that she had the condition, so she passed it on. Okay? Now, if she has a son, her X chromosome, right, she has two X's. Theoretically, one copy of all of her genes will be passed to that son. Okay? That son has the Y from the father. So if she passes the bad gene, that's hemophilia, onto the kid, he has it, and it's expressed in the phenotype because he's male. If she has a daughter, then she might pass on the bad gene, but the daughter has a second copy from the father's X, and therefore she would just be a carrier because it's recessive, basically, is the answer. So do you understand now? Yes. Okay. So that's hemophilia, and that's inheritance, and that's how this works, and that's what sex-linked traits are. Okay. And that's why people say things like, oh, baldness skips generations, because the generation it's skipping is the mother, right? The mother's father has it, the mother's son has it too. It skips a generation. Well, it's really about being sex-linked more than it is about skipping generations. Okay? Yes, sir? There was no evidence that it existed prior to that, yes. So, I mean, it's something that I think they're relatively certain about, but perhaps it's not considered a known fact, but a strongly supported hypothesis. That's yeah, a lot of, notice that nobody has it now. You know, poor William and Harry are in the clear and can frolic and play nude billiards all they want and still not have to worry about getting hemophilia. Yes.
Except for Leopold, they were all passed down by one
Leopold was passed down by women too, because Leopold is the son of Victoria. So yeah.
I think she means like the offspring of his, right?
Oh, I see. You mean like Leopold has offspring who inherit it from Leopold. I don't think the died in infancy children are actually listed on here. What's that?
So Leopold passed it to his daughter, and she ends up a carrier, and his son didn't get it because he can't pass it.
Yeah.
Okay, got it.
Right.
No, he can pass it.
He can't pass it because he has it on his X. So he can pass it. If he has a daughter, then he's going to be passing his X to the daughter because the daughter is 2 X's, and so she has to get one from him.
He can't pass it to them.
Not to his son.
Yes. Not to a son, yes. But he can to his daughter. Yes. Yes. Got it? Okay, good. Nice. All right. Oh, my God. This is so complicated, I can't take anymore. Okay, this is what Tegrity is for. If you have no idea what's going on, you can review this lecture later. You can go to sleep with me telling you stories. Last but not least, and this is not an exhaustive list, but I'm trying to hit some highlights here of things that don't follow Mendelian patterns, are environmental influences, that is to say non-genetic factors influencing the phenotype. Okay? Here is a picture of me after 3 minutes in the sun. Okay, I stuck sunburned douchebag into Google, and that's the guy that came up. No, I didn't. I'm kidding. But that's just a picture of somebody with a sunburn. Okay, that is a non-genetic factor that influences the phenotype. As we've seen, there is a genetic basis for the production of melanin, which this person does not do a lot of. But the sunburn is the environmental influence of being exposed to the sun. Okay, here is another example. Know what that flower is? Hydrangea, nice. How many people in this guy's in purple? Oh my God. You know, it's students like you that make writing tests so difficult. What do you mean purple's right? I used to have a question in the primate assignment. Did I say this? I have a question in the primate assignment that said something like, where is your primate found? Right? And half the class would put in the trees, and the other half of the class would put on the face of the earth. Right? And it'd be like, okay, can you be a little bit more specific? So now I have to say, what is its geographic range and habitat? Please list countries in order of you know, how common it is. Anyway, yes, it's purple. Yes.
If it's all multiple choice, then
only one answer can be right. Yes.
Yeah, I know, but how do they feel on that?
It's not a multiple choice. It's an assignment. It's not the test. Yeah. That wasn't a test question. It was an assignment question. But that's
They still got me scared on the test thing. So.
Yeah, good. Be very scared. What's that? Yeah, so the question is, in addition to identifying the flower, which is really not the problem here, how is this a non-genetic factor influencing the phenotype?
That's because the soil is acidic.
Yeah, well, it becomes blue when it's an acidic, and it becomes pink. It's blue acidic, yeah.
Oh, okay.
So you can change the color of the hydrangeas in your front yard by planting pennies in the ground in between flowering seasons, and you can change it from blue to pink. That's pink, yeah. That's pink. It's not the best color, but I mean, as far as the photograph goes. Okay, didn't know what a hydrangea was? Here's your chance to make up for it. What's this? Red. It's red. No, the answer is red. It's not a bird. It's red. Okay. Why don't you go do that with your weekend? Yes, please do not stick a penny up the ass of any creatures at the zoo when we go on our extra credit trip. Yeah. Listen, laugh it up, smartass. But I will tell you that thanks to somebody feeding a penny to a gibbon at the zoo incurred like some $20,000 vet bill because it was going to kill it by poisoning its liver after somebody tossed a penny to make a wish into its cage, and the thing swallowed it and had to be operated on. Yeah, it's bad. You also can't make out with gibbons because they're highly susceptible to virtually every human sexually transmitted disease. Totally. Herpes can kill them in 48 hours, in fact. They don't. Yes. Yeah, and flu can too, and so everything can. Chicken pox. So when you're sick, yeah, you just have to be. If you're sick, you can't go to the Gibbon Center. No, the distance is what makes them susceptible to diseases. So they're further away from us. Yeah, there's 20 million years between us and them. Yeah, but you'll have another date. The Gibbon Center dates haven't been set yet. It's okay if you have to be sick with, something. A cold is okay.
Something bad.
Just don't make love to them. They now put up barriers around the cages, so the gibbons are probably safe from your amorous advances. All right. We need to keep moving here, as amusing as all of this is. What's the non-genetic factor influencing the phenotype in this creature? Yeah, what's the diet? Yes, so the more they eat of shrimp in their diet, the higher amount of carotenoids they get, which is responsible for red color throughout the animal kingdom. We had a consistent problem with this actually when I worked at the fish store, because the food that they manufacture for tropical fish is not high enough in carotenoids. So people would buy like fish that were red in color, and they will fade over time, actually, consistently. And it's not just fish; it's also like crustaceans. Anything with that red color in the ocean gets it from carotenoids. So if you give this guy, if you start feeding him Big Macs, he'll get lighter in color. There's another project for you. What's that? Yeah, and then they get green. They get green. They get red over time. Yes. Yes. It does affect us slightly, although not quite as much as the flamingo. Last but not least, don't know what the flamingo is either. What's this? I'll accept cat. Thank you. It's what type of cat? Siamese. Okay. And what is the non-genetic factor that's influencing the phenotype? Love it. Wow, you guys are kind of smart. Maybe you will pass the test. No, I don't think so. It is temperature, because basically, like other creatures that we've talked about, the dark color on the cat is produced by melanin, but the melanin is denatured by another amino acid. And that amino acid causes it to denature and lighten the color of the cat when the temperature reaches a certain point. And therefore, it is the warmest regions of the cat that are lightest in color, and it has its dark color on its extremities, which, if you think about it, is actually a pattern that's pervasive among, say, cats and dogs. You can do an experiment. Take your Siamese cat and put it in the freezer for a few weeks, and you will see that it gets much lighter in color. Actually, if you want to do something more humane, you can put little booties on its feet, and the feet would get lighter in color, actually, fairly quickly. Or if you have, does anybody have a Siamese cat? Nobody? Okay, so if you do, but they do get darker in the winter, and that's why. That's what's going on. Your neighbor. Okay. Congratulations, but if it's just your neighbor's cat, I can't give you any extra credit. Had it been yours, I would have given you an automatic A. Yeah. Get away from that cat.
Darker with winter?
Yeah. Warmer is lighter. Warmer causes the melanin to denature. I'll show you the amino acid when we get to it. All right, that's...
The melanin, is incorporated also into their fur, not just their skin?
Yeah, it's deposited in the fur like it was for the Labradors. Okay. Yeah, same deal. Yeah. Put socks on your cat. You'll see it happen.
They walk funny.
They walk funny. Yeah, they're kind of difficult creatures. What?
Would age be a non-genetic factor when that influences the phenotype?
What we call aging would be both. A lot of what we think of as things are actually influenced by both. I mean, think about like a sunburn that's influenced by the genetic basis for the production of melanin, but also the sun. So a lot of things, one of the things that I want you to take away from, genetic versus non-genetic factors in the phenotype is that generally speaking, it's a combination of, it's not nature versus nurture, right? And there's a lot of evidence to suggest that many genetic triggers or many genetic factors have to be triggered by some sort of exposure in the environment to certain things that cause them to be triggered. And this turns out to be a very important area of study in genetics called evo-devo, which looks at the developmental exposure to different things like hormones, right, which influence actually how a fetus develops and what you become as you grow. So essentially, there's a famous study that shows that the more sons a woman has, the more likely the later sons in the birth order are to be gay. And the reason is, and this has been replicated multiple times with multiple populations, and the reason is that there are hormone changes in the woman that happen by virtue of having given birth to kids before that change the hormone exposure during development in the son as it's developing in her that makes the kid more likely to become gay. Right? Now, of course, gay is a behavior, so it's not just like something that's automatically genetic, but it appears to have this genetic basis that increases in likelihood. So you ask yourself the question, is that genetics or is that environment? Well, the exposure to the hormone in utero is environment. It's the utero is environment. So what this actually shows, and we'll talk a little bit more about Evo-Devo at the end of the next lecture, probably actually on Wednesday, but what it actually shows is that Lamarck wasn't entirely wrong. Because during the developmental phase, when something is, a fetus and developing internally to the body of its mother, human or not, that certain things happen environmentally that actually affect which genes, the transcription factors, like the switch genes that make the dog get blonde or not, right, yellow or black labs, those will switch on and switch off during development. Most of those regulatory genes are set, that is, switched on or switched off as part of your development, right? After you become a zygote, after the egg gets fertilized. And those switches are all set at that point. Well, those switches are set partially because of what happens environmentally, internal to the body of your mother, right? So if, for example, your mother is like my mother and smoked like a chimney and drank like a fish through her entire pregnancy, you end up with having a lot of problems like me. You end up being a crazy person like I am, right? And the funny thing is that I always said this as a big joke, but then now it turns out that there's a fair amount of evidence mounting for this sort of thing. Because those switches get set during that particular point. It can happen during just your life and exposure to the environment as an organism, but the development is so critical when those regulatory genes get turned on and off that's really what people are interested in now. It also relates, of course, to genetic engineering. So this is a very critical interest of research, area of research interest right now is this, it's called evolutionary development, or Evo-Devo for short, or epigenetics, it's also called.
Say, like, it switches on and off, like, more sensitive when you're still.
You're developing, that's why. So your most critical development happens during that time. Now you can say, oh, you're still developing now, right? So yeah, you might have a genetic basis that gets triggered for something, by environmental exposure. Maybe you end up with sun cancer because you get too much sun or something like this. But really, it's the in utero phase that's really critical. And I can refer you to a book on this topic if you're interested in it. I'd love it if you'd write your paper about it, actually.
So, this studies were all done on women that had two boys or just a mixture?
Are you talking about the gay study? The gay study was conducted on women who had multiple sons, and what the studies all show was that the statistical likelihood of them being gay increases as they are later in the birth order.
Twins too, right?
The correlation does not hold for girls. The birth order correlation has not been reproduced, and they've attempted multiple times, but they've done 5 or 6 versions of the boy study, and it's correlated every time.
So then, what would happen? Is this for women that carried the pregnancy?
Yes. Yeah, I believe that's all they selected for, yeah. I'm pretty sure.
They were aborted prior to.
Make that your master's thesis. I have no idea. It's kind of gruesome, but yeah, you would wonder. That would actually be the next logical question to ask, because if you're really talking about something that's a hormone cycle that's impacted by pregnancy, I think it's really impacted by carrying fully to term and actually experiencing birth. I think that's right.
There's a hormone level that are interrupted not to term, so then that.
Yes.
Because my mom had like 3 boys before me, but then aborted them all, and then I came along and I'm the firstborn, and hi, I'm gay.
Well, maybe it holds for it. I don't know. I don't know the answer to that question. It's a very good question.
Yeah.
Right. So there would be a hormone effect of that. The question is whether or not that's strong enough to produce the effect that they're saying. And it's not like some sort of guarantee or something. It's just a statistical likelihood. Yeah. I think they've eliminated that. I can give you references if you're interested in reading more about it. Yeah, I think that it's really just about hormone exposure in the process of birth order, and it's not a genetic basis. The genetic bases for things like homosexuality are not well supported by research. They can't narrow it down. They can't say, oh, there's this gene, it's right there, and that's what it does. But this birth order result has actually been well supported. Very interesting in the last few years. That's just one example. There's a lot of other stuff in this area. Anyway, let me move forward, you guys, and we'll get through the material. My goal is to get through a bunch of the genetic stuff today, and then we will finish it up and then watch a film on Wednesday. And then you'll have the test. This is the one we want. Shall we start with a video? Let's start with a video. Here we go. It's 3 minutes long. I know your attention span is shorter than that, but you'll just have to. Let's see. All right.
Genetics is a discipline of biology and is the science of heredity and variation in living organisms. The fact that living things inherit traits from their parents has been used since prehistoric times to improve crop plants and animals through selective breeding. The modern science of genetics seeks to understand the process of inheritance, which began with the work of Gregor Mendel in the mid-19th century. Although he did not know the physical basis for heredity, Mendel observed that organisms inherit traits via discrete units of inheritance, which are now called genes. Genes correspond to regions within DNA, a molecule composed of a chain of 4 different types of nucleotides. The sequence of these nucleotides is the genetic information organisms inherit. DNA naturally occurs in a double-stranded form, with nucleotides on each strand complementary to each other. Each strand can act as a template for creating a new partner strand. This is the physical method for making copies of genes that can be inherited. The sequence of nucleotides in a gene is translated by cells to produce a chain of amino acids, creating proteins. The order of amino acids in a protein corresponds to the order of nucleotides in the gene. This relationship between nucleotide sequence and amino acid sequence is known as the genetic code. The amino acids in a protein determine how it folds into a 3-dimensional shape. This structure is, in turn, responsible for the protein's function. Proteins carry out almost all the functions needed for cells to live. A change to the DNA in a gene can change a protein's amino acids, changing its shape and function. This can have a dramatic effect on the cell and on the organism as a whole. One of the most fundamental technologies developed to study genetics, DNA sequencing, allows researchers to determine the sequence of nucleotides in DNA fragments. Developed in 1977 by Frederick Sanger and co-workers, chain termination sequencing is now routinely used to sequence DNA fragments. With this technology, researchers have been able to study the molecular sequences associated with many human diseases. Researchers have sequenced the genomes of many organisms, using computational tools to stitch together the sequences of many different fragments, a process called genome assembly. The large amount of sequence data available has created the field of genomics, research that uses computational tools to search for and analyze patterns in the full genomes of organisms. Genomics can also be considered a subfield of bioinformatics, which uses computational approaches to analyze large sets of biological data.
All right, did you get all that? What? All right. Yeah. Isn't it already on there? I think I put it on. I'll double check. I thought it was on. It's in Integrity now, so it's fine. It will actually help you if you did have trouble following the density of information, particularly when they were talking about protein biosynthesis, which is what we'll talk about today. When we get done going through the material, if you re-listen to it and you've sort of digested what we're covering here, you should be able to follow the video much more easily. So I would give it another watch after we finish the lecture and see if you can't follow it more effectively. I'm sure you got some, I know you got some things out of it, and that's why I show it to you ahead of time, but I don't expect you to sort of remember everything you heard because it's a pretty quick crash course. But I think by Wednesday you'll be in better shape. Okay, so if I want that one. Yeah, it's because it has a split display on the thing, so it asks every time. All right, so molecular genetics is our topic today and will be our topic for the next couple of days. It'll take me the rest of today and a good part of the next class to cover it, and then we'll end with a film that looks at some recent research within biological anthropology on topics related to molecular genetics and inheritance. A few things to talk about. First of all, if you have your genetics key terms with definitions handout handy, some of the things that I'm going to cover are on that handout, and you don't necessarily have to write them all over again. You can if you want, but you don't necessarily have to, so have that nearby. Let's see. I'll dump that one out. Put that one in. Okay. Cellular organisms can be divided in a number of ways on the basis of a number of characteristics, but a very basic definition is between what are called prokaryotic, sometimes called single-celled organisms, and eukaryotic, which are multicellular organisms. But not always, yeah. It's a little bit of a slippery thing. There's a couple of things that you should understand from this without getting too deep into this topic. Number one is that humans are eukaryotes. That's one thing you should understand. Another is that eukaryotic cell structure involves cells being what's called nucleated, meaning that there's a nucleus in the center of the cell. Now, if you've studied cells like in bio, you were probably studying eukaryotic organisms. So in your brain, if I say, what's the structure of a cell, you're going to go, oh, there's a nucleus in the middle. That's not true for prokaryotes, though. They're simpler in their cell structure. This is a critical, actually, topic in the history of evolution. And there's a famous evolutionary theorist who's a researcher called Mark Ridley. And he asserts that the moment in evolutionary history when complex cell structure arises, right? So eukaryotic cells are more complex structurally, okay? They, from all evidence that we have, evolved from prokaryotic ancestors that were simpler, things like bacteria, for example. So what Ridley asserts is that the evolution of complex cellular structure when eukaryotes evolve is actually, as an event, even less likely than the original evolution of life on Earth biochemically, which we know a bit more about at this point. Now, it's entirely conjectural. I mean, he's spent his life studying this, and he's kind of brilliant. He's not stating that this is a fact, but he's merely trying to emphasize that the complexity of our cell structure, in his opinion, is sort of like the spark that enables the rest of higher order organisms to evolve. This was necessary for that to occur, in his opinion, and an extremely significant event. One that, in his opinion, is less likely than the biochemical evolution of life originally. So, but whether he's right is another question, but it does sort of underscore the significance of this division.
What about the endosymbiosis theory put by Willis? I forgot her first name.
Lynn, yes,
Willis?
yes.
Yeah.
Can you tell people what it is?
Basically just saying that you have one prokaryotic cell engulfing another prokaryotic cell, a smaller prokaryotic cell.
Yes. And that's what produces eukaryotes.
Yeah, essentially. And then it goes on. And then
Seems like as likely a scenario is, I know this is debated, right?
it is, but I mean, the mitochondria in our cells now have
Support them.
S ribosomal RNA, it
Yep.
has double membranes.
Yeah.
Everything about it supports that it was at one time.
Its own independent organism.
Yeah, plus
Yeah.
it also divides independently of our own cells.
Yes. Right. Yeah. I think that what Margolis has is sort of this, the most likely explanation for the sort of unusual place of mitochondria. Which actually, why don't we return to this question, because I have sort of a cell picture coming up, and then everybody else can kind of be a part of the discussion too. Okay, a couple things about human cell types. Some of this we've learned, but this is terminology. Somatic cells is a general term that refers to any cell that forms the body of an organism. Soma means body. So somatic cells are everything other than gametes. Gametes are reproductive cells of sperm and egg in animals. A zygote is a fertilized egg. Gametes we've covered. I don't think we've talked about zygote. Zygote is on your key terms list with definitions. Fertilized ova or egg, it divides by mitosis and becomes an embryo, ultimately becomes a fetus, ultimately becomes an organism in a reproductive event. So those are just three basic terms to be familiar with. I think the last two are on the handout. Is somatic cell on the handout? No. I can't squeeze any more on that damn handout. It's going to go to 3 pages or 2 pages. It's good for you to write it down. Shut up, kid, you bother me. I mean, you're not as bad as the guy that said purple for flower, but you're getting there. What? Huh? I'm just going to call you purple from now on, actually. All right, am I good? Can I go forward? All right. I told you that if I tease you, it means I like you, right? That's like the messed up family I have. We can't express affection. We can only say mean things to each other instead of saying I love you. So every mean thing I say to you really means I love you. All right. So here's the picture of the cell. Now, this is, of course, a eukaryotic cell. Here's the nucleus. The nucleus is known, obviously, by most people who are familiar with this information as containing the DNA for the organism itself. Okay, so inside the nucleus of your cell is a little blueprint of you. Okay, and for people who have trouble sort of getting how genetics works, which is like any normal human being, there is a popular science writer who wrote a book called Genome, which has 23 chapters, one for every chromosome humans have. And his name is actually Matt Ridley. He's no relation to Mark Ridley. And Mark Ridley is like a real scientist, but Matt Ridley is like a popular science writer. He writes books for normal people to read. So he wrote a book called Genome, and he asserts in the book that the best way to understand genetics is to use an analogy of a book. That's why he put the book in 23 chapters. So basically what he says is that the best way to understand genetics, or the genetic code, as your little video called it, is to think of the book as being the book of you, or the blueprint for you. Right? So inside the nucleus of your cells is an instruction manual as to how to build you. All that genetic information is contained in that nucleus. Okay? So that's what we call the book. Okay? So basically, he claims that through a kind of language metaphor, anybody can understand how this whole thing works. Now, it is a metaphor, but it's sort of a way to think about genetics that will help you kind of put it together and understand how all the pieces fit. Okay? So basically, the language that the blueprint for you, or the book of life, or whatever you want to call it, is written in is called DNA. Or we could call it DNA-ish, right? It's English, Elvish, Spanglish, DNA-ish. Okay? That's the language. We'll get to the rest of it. We'll fill this whole thing out as we're talking today, and it'll help you kind of understand things. Now, this is not a cellular biology class. I don't want you to know too much about cell structure, but there are a couple of little pieces of information that are critical in understanding what we're covering. So you don't need to write all this down. I don't care about all of this. You do need to know that the nucleus contains the genetic material. There are a couple of other cell structures that I would like you to be familiar with because they have particularly important roles in bioanthropology. One is the mitochondrion, which is what Justin brought up. And that is called an organelle. And organelle is spelled like this. And it basically produces energy for the cell. And the DNA evidence for mitochondria is the plural, mitochondrion is the singular, is that it was its own independently existing organism in evolutionary history because it has a highly simplified DNA of its own that is inherited not with the rest of your body's somatic DNA, but independently through your mother. And the fact that its DNA is very simple makes it quite useful in the study of population genetics. This is how it becomes important for us in anthropology. Basically, like your somatic DNA, when reproduction happens, errors occur. Okay? The errors in your book we could call typos. Those typos are mutations when little mistakes happen, okay? And everybody's book has some typos in it. Actually, every human generation leads to approximately 50 mutations. We all each have that in our genome. Most of it does nothing because it's junk. But everybody's got these typos in their book. It just happens. The proofreading process isn't perfect, okay? And the fact that it's not perfect is critical to evolution because mutation produces new variation, and that's important in the whole picture. So, mitochondrial DNA, and we'll talk more about it in the future, is inherited through the mother. It's very simple. What it's useful for is that it has little mutations in it too, and if you look at the DNA in mitochondria closely and you look at the mutations that, say, make you and me different, you can actually figure out with a good deal of accuracy how closely we are related. So you and I will have more mutations in common if we both come from the same culture or the same part of the world, this kind of thing, or the same family even. There's even family-level mutations. So people use mitochondrial DNA to figure out ancestry of human population groups because you could do the same thing with somatic DNA, but you have 3 billion base pairs, whereas mitochondrial DNA is less than 20,000. So it's easier to do it with mitochondrial DNA. And thus, when you have a company like, let's see if I can do this here. When you have a company like this company, which is African Ancestry, and if you are an African American person, you may have no idea where your ultimate family ancestry lies because that information would be lost during slavery. So what you can do is look at your genetics through this company, and they will line up your DNA with all the information they have about different lineages from Africa and basically tell you where your family comes from, what area, what ethnic group in Africa. And a lot of famous people have done this. Oprah did a show on it where she did it, but many people have done it because that information is lost to them. Anyone can do this, but there's actually several companies that actually specialized in African American ancestry. And this information can be accessed through your mitochondrial DNA. Because that's so much easier, having this done is really pretty cheap. Okay, it doesn't have a 100% accuracy rate, but they're getting more accurate as time goes by because techniques improve. If you want to have a full medical scan of your DNA done through a site like DecodeMe, you have to pay about $1,100 for them to look quite closely not at your mitochondrial DNA, because that's not going to tell you much about whether or not you have a genetic likelihood toward pancreatic cancer, but rather they'll look at your somatic DNA, which is a lot more involved, takes a lot more work, and thus this costs $1,100 and the other site costs about $100 or a little bit less. And that's why mitochondrial DNA is used partially because it's just easier. It's the genome of the mitochondria is so much shorter than your somatic genome. Yes.
So the tracing your ancestry from mitochondrial DNA wouldn't that be tracing your mother's side?
Yes, it would. Yeah. So if your parents come from opposite sides of the earth, it's only telling you one part of your heritage, right? Which can be a problem, right? I mean, it can be a problem in the sense that it may render the information useless depending on your particular family history. Yeah. But you can also use, I mean, if the mitochondrial DNA method failed you because, say, you know, your African American heritage comes from your father's side or something, then you could use other methods to get the same information. It would just be more detailed and difficult. Yeah. Please.
So when they do paternity tests, they have to do they can't do it for mitochondrial?
No, they use somatic for paternity tests. Yeah. Paternity tests are somatic, for sure.
Is mitochondria like a prokaryote inside eukaryote?
Essentially.
That's what I thought.
It has that history. Yeah, that historical trace is there, if you think of it that way, yeah. So anyway, there's a couple of websites for you. They're interesting. It's not a bad idea. In your lifetime, I think, you know, it's going to become, let's see, meth lab. Love that. Why does that give me such amusement and pleasure? In your lifetime, genetic testing is going to be greater and greater in its significance, and it'll be, you know, as techniques are refined, the use of somatic DNA will be the norm. It's just, you know, you got to wade through so many base pairs to get to the information. There's certain areas, like, you know, if you're going to use the mitochondria to determine who's more closely related to whom, you want to look at the stuff that's different. We all have a bunch of stuff in common, but who cares about that? That doesn't tell you anything. We all have it. What you want is to look at the stuff that's different. So when you do these kinds of population genetics studies of different humans, you're trying to determine who's most closely related to whom, you actually focus on this area of the mitochondrial DNA that's called the D loop, which is where the high rate of mutation occurs. And the high rate of mutation occurs there because it's junk. Basically, if you're talking about junk DNA, all the mutations in the world make no difference at all. So they might as well accrue like crazy because it's not going to cause you any problems at all in the shape of your mitochondria or your ability to learn in this class or whatever it is. So we accrue the most mutations in our junk DNA because they don't do anything to us, so we just pass them on to our offspring. Right? So that's what you end up looking at. You're actually interested in the junk when you're trying to determine things like ancestry for that reason. We'll go over this again if this isn't all clear. Okay, so back to basics. Structure of DNA, double helix, twisted ladder, flattened for this overhead. Yes?
Sorry, just back to the basically all we have to do is just better primers, right, for PCR?
Yes, yeah, basically. And that's happening, and it'll get better and better. By the time you guys are old and dead, you'll be amazed at what people can do. Yeah, it's already happening. I mean, these techniques, the error rate for example, determining ancestry is much lower now. I mean, it's much more accurate because, I mean, just in terms of these companies have been around for like 10 years, and they're so much improved. When these companies first came out and were using mitochondrial DNA, they had, you know, it was like a 60 or 70% accuracy rate, which is just horrific for science. I mean, you know, what is that? That means you've got a likely guess, but, you know, you want to do better than 60% on the test, and you probably want a company you're paying at least $100, if not $1,000 to have a higher accuracy rate as well. Oh, guess what? You're going to have pancreatic cancer, but it's about a 50-50 chance. Like, oh, thanks. Can I have my money back?
How inaccurate, though, I mean, in terms of, like, they're doing geographical locations, right, where your
Yes.
ancestors are from?
Yeah.
So, I mean, were they kind of in a neck of the woods or completely off?
No, they're pretty good. They're pretty good. They're pretty accurate. I mean, at least can get.
I'm talking about when they were inaccurate. I mean, if you're.
Oh, well, see, part of their accuracy came about as they built up their database and can compare the different genetic sequences between the people who have used their services. So that.
What they had, they were doing.
Yeah. So basically, and so now that they've had more clients, that in itself increases their accuracy too, because they can compare. You know, like if I walk in and I have exactly the same DNA sequence for comparison that you did, it's a no-brainer that you and I are related to one another. And they do what's called lineage coalescence. Like you get that information for thousands of people, then you start to build a family tree, basically. And that increases their accuracy, too. So the longer the company's around, the better off you're going to be. It's a little bit different with the do you have pancreatic cancer issue. Because, you know, things like cancer, again, are going to be a function of not just a genetic predisposition, but also environmental exposure. So to say, like, okay, you've got a gene that makes you predisposed to getting breast cancer doesn't mean you're automatically going to get it. Maybe you can help avoid it with diet and exercise, right? So these tests are never absolutely going to be conclusive. In some cases, they'll be more conclusive. They'll be more conclusive than others. Like, so for example, some genetic mutations, virtually ensure that you'll end up with a particular genetic disorder. When we get to population genetics, I'll show you a short video on Huntington's disorder, which is a genetically based mutation. And if you've got that genetic mutation, you are virtually certain of getting Huntington's. It's determined, right? They call that heritability, right? So heritability, which we can write on the board, heritability is the percentage of variation that's due to genetics. It's one way to define it. So if you have 100% heritability, that means that you are virtually assured of getting a condition if you have the gene for it, right? So a lot of the studies of various genetic disorders yield heritability numbers that are more in the 50% range. So, it turns out that the heritability of, the heritability, here's a good example, because you brought up twins when we were talking about homosexuality. The heritability of homosexuality in twins is 50%, more or less. There are different studies that have given different results. But that means if you have identical twins, the likelihood that if one of them is gay, the other one will be is 50%, which means that's very strong evidence that it's not genetically determined. It is partly, but not totally, right? And I'll tell you, in our society, there's a real tendency for people to embrace genetic determinism. People really think that, like, who they are and what their destiny is totally controlled by genes. But that's, and I understand that, so. Well, there's that, but then, if you look at, no, but I completely agree. Yes, I think people use it to shirk responsibility. Oh, it's my genes. I have no control over what I'm doing. It's my genes. That's what turned me into like a multiple murderer and a sexual abuser. It's like, okay, great. But if you think about something like, gay marriage or something, right? I mean, if homosexuality is a genetic thing, then how do you make the case that it's a choice and therefore I should be able to restrict your marriage rights or something like this, right? Because the case against it is based on this notion that it's a choice, and it's just like, well, what does the scientific evidence say? Does it say that it's a choice? It doesn't say it's a choice, but it also doesn't say that it's automatically controlled by one genetic locus or anything like that either, right? At least the heritability studies tell you that it's not simply nature, but nature and nurture. And that's kind of normal, I think, right? It's more normal. Genetic determinism is a simplistic way to think. All right, back to this. So here we have the structure of DNA. And DNA is like a ladder. It's like a little ladder. And the rungs of the ladder are composed of base pairs that occur in predictable complementary oppositions. C's, G's, A's, and T's. C's and G's are, they're bonded together. And A's and T's are. The bases, these are called bases. They occur in pairs, so we call them base pairs, are adenine, guanine, thymine, and cytosine. You don't have to remember those names. It's fine. If you can remember C's, G's, A's, and T's, I'm thrilled. The sides of the ladder, which form the structure of it, are phosphate sugar bonds. The combination of a phosphate sugar bond with a base, it's called a nucleotide. Okay? So, basically, if our language is DNA-ish, our alphabet is C's, G's, A's, and T's. We'll take heritability off of here. So that's going to be our alphabet. Well, the problem with an alphabet that only has 4 letters is that words take a lot of letters to spell. So it takes a long time to spell out information if you can only use 4 letters for contrast, right? So basically, genomes are extremely long and complicated for the most part. Okay? Some facts about DNA. It is a large organic molecule. It stores the genetic code. It is composed of sugars, phosphates, and bases in a double helix-shaped molecular structure. Your genome, the book of you, has about 3 billion base pairs. That sounds very impressive, but it's actually not particularly long for organisms on Earth. The longest genome is about 6 billion base pairs and belongs to an amoeba. plants, so it's not about complexity. It's not, the length of the genome does not necessarily correlate to the complexity or order, like we talked about higher order organisms like animals and humans. Plants, because of the way they reproduce, have a lot more junk DNA than animals typically, and their genomes are quite long. The shortest genome of any vertebrate belongs to a pufferfish. If you went and saw Nemo in 3D, you might remember that there's a pufferfish in the aquarium, the one that puffs up and has spikes on it. There is, in fact, an entire research project, an international effort composed of scientists from all over the world studying the genome of the pufferfish simply to understand why the hell it has so little junk DNA. Right? Go figure. You got plenty of junk. You got plenty of junk in your trunk, baby. All right. Segments of DNA and chromosomes correspond to specific genes. The little film I showed you made this point, too, and we'll get back to genes. But we have to do some other stuff before we get to genes. You already know that gene means many things because a regulatory gene is quite different than other types of genes. So the word gene has a really broad number of meanings and applications. But we'll get back to that. I love that the company is called Decode Me. Decode Me. It's sort of self-centered as well. Decode Me. All right, so when cells divide, DNA replicates, and it's able to do this because of the complementary nature of the base pairs. So it can unzip, as you've probably heard, shown here over on the left, and what happens is that free-floating nucleotides in the nucleus of the cell will line up and bond with each side of the unzipped DNA and form two new strands. Okay? This happens actually extremely quickly. It looks really complicated on an overhead, but it happens really fast. So that leads us to cell division, which comes in two forms. And I don't, if you take this class from my office mate, whose father is the chair of the botany department at Cal State Fullerton, you get a much lengthier and detailed explanation of mitosis and meiosis. Mine, however, will be much quicker. Let me say a few things. First of all, scientists are terribly fond of using two almost exactly sounding like words to describe two different processes just to confuse you, particularly if you're slightly dyslexic like I am. So mitosis, this is what you need to remember first off. Mitosis is the process of cell division for somatic cells, and meiosis is the process for gametes. Okay? Note that the division as it occurs, which utilizes this replication of DNA in the case of mitosis, results in two identical daughter cells that each contain the same number of chromosomes that existed in the original cell. Meiosis, however, since it's producing gametes, is a double division that results in cells that have half of the genetic complement of their original cell. So there's only 23 chromosomes in each of them. Now, one of the points that I want to emphasize, where is it? There we go. Well, actually, one of the points I wanted to emphasize for you is donuts. This is cell division as shown in donuts. I thought this might help you remember it for the test, since donuts probably make a bigger impression on you than my other overheads. Or my personal favorite illustration of cell division, which is, can you explain cell division very fast? ... Thanks. Right. Little screen grab. Yet another treasure from I Fucking Love Science on Facebook, which is one of my favorite friends. Now, basically, you have, and we'll talk more about chromosomes in a minute, but humans have 23 pairs of chromosomes. So they're all paired, and the same genes exist on the same locations or loci on each of the chromosome. We talked about this before. So you have, when cell division occurs, the DNA material in the nucleus of your cell organizes into the chromosomes. They are paired. Here, let's say, is chromosome 5 for the hell of it. And you've got your A member of the pair and your B member of the pair. So you're going to have 2 copies of each of your different genes, one here on A, one here on B. They'll be in the same location on each of them, and they mirror each other. The word for this is homologous. Okay? So they mirror each other. Now, what's going to happen when you produce your sex cells? Okay, when you produce your sex cells through meiosis is that you're only going to be donating half of that genetic complement to your sex cells to produce offspring. But it's not that you donate all of A chromosome 5 to your little sperm guy, okay, or your egg, right? Or all of B, right? And it's not like you do 5A, 6B, 7A, 8A, 9B, right? What happens in reality is that they break apart and you get little pieces of each chromosome. And this makes sense because if you were donating, think about the advantage of sexual reproduction that we talked about when we saw the film. It's that it creates variation among your offspring. If you were, if meiosis resulted in all of one chromosome or the other in a pair of them being passed on to your gametes, the possible results would actually be rather limited because you'd either be either A or B. But since they break apart into little pieces and come back together again, the possible results are virtually infinite. Okay? So in other words, the fact that they break apart and come back together again, which is a process we call recombination, is absolutely critical in the advantage of sexual reproduction that we talked about before. Okay? So what this illustrates is that you've got 2 chromosomal configurations. They break apart in the meiosis. In meiosis as a process, and what you can end up with is either, in this case, you've got one dominant gene and one recessive, or the other way around, one recessive and one dominant. You've got all these possible combinations, even though originally it existed like this. So recombination we define as the exchange between homologous chromosomes. It occurs during meiosis and the production of sex cells. Chromosomes break apart and recombine during that time. This sounds like it's insignificant, but I promise you it's absolutely critical to why sexual reproduction is helpful for higher order organisms. It produces a novel set of genes for each individual and an infinite number of possibilities. However, genes that are located close by one another on the same chromosome are often inherited as a unit. That's called linkage. And that's why you get brown hair and brown eyes together, or why my niece looks like a little white girl and my nephew looks like a little Japanese boy. I told you that, right?
Yeah.
Yes, okay.
So you can carry some, like some people have like special needs children and some of their kids aren't.
Yes.
You can carry something in your DNA and not give it to every single one of your children.
Yes, because of the fact that you're only passing down half. So the thing is that since you only pass down half of your genetic material and you've got 2 copies of everything, part of this whole thing is a total chance crapshoot. Yeah, totally. Absolutely, yeah. So there's this 50-50 thing. Which of your 2 genes are going to pass down? Well, if you're homozygous, you've got 2 copies of the same thing. Doesn't matter. But if you're heterozygous, then you could end up with completely different situations.
Certain traits that both parents have, that's just happen.
Certain traits.
Like certain, if both are like have dominant traits.
Yes.
That will happen no matter what.
Yes. Yes. So the segments of DNA that are inherited together as part of linkage are called haplotypes, and that notion will come back up in the class in a moment, or actually next lecture, maybe after your test. All right, that leads us to protein biosynthesis, which is about the production of proteins at the cellular level. Now, the information that's contained in DNA does not leave the nucleus, but the information can be taken out of the nucleus by RNA, which comes in several forms. In order to produce proteins, that information in the DNA has to be taken out of the nucleus and taken to the ribosome, which is written there. And please know that. If you want to put that in your notes, it'd be great. That the second phase of protein biosynthesis, known as translation, occurs in the ribosome, is what I would like you to be familiar with. The ribosome is part of the cell out in the outside of the nucleus, past the nuclear membrane, in the area called the cytoplasm. The cytoplasm is this whole sort of blue area here. The ribosome is located there. So in order to produce the proteins, you've got to get the information from the DNA out into that ribosome. That's going to be accomplished through a two-phase process known together as protein biosynthesis. The two phases are called transcription and translation. Transcription occurs in the nucleus. Translation occurs in the cytoplasm. I have worked quite hard in order to be a happier teacher with happier students to find, I literally have scanned textbooks to find the simplest illustrations of some of this stuff so you can just see it most easily. This one's from your book, which actually has pretty good illustrations. But my favorite, simplest protein biosynthesis illustration of all time, which isn't quite as easy as the donuts, but is coming up, and it's not in your book, actually. Okay, so transcription's phase number 1. Definition of transcription is the formation of messenger RNA, which we abbreviate mRNA, like this here. Again, this is formed from free-floating nucleotides in the cell's nucleus, much as DNA replicates in the process I showed you before, where it unzips. In this process, thymine is replaced by uracil, but I'm not asking you that you remember this, so it's okay. You just understand that the messenger RNA has a slightly different structure. During this time, the non-coding sections of DNA, which are called junk or introns, are removed, and the coding sections, which are called exons, are the only ones that undergo transcription. Transcription is the first of the two steps in protein biosynthesis. At this point, once the mRNA has been assembled, it will then pass through the cell membrane out to the ribosome where the second phase occurs. The exons are the coding sections of DNA. Yeah. If this doesn't appear significant to you yet, it will. Why it's significant is coming up. It's really the production of proteins, which serve many functions in your body, and they're critical. That's why it's sort of critical to understand those. Does that mean it's 3? Gosh, we're almost out of time. The next little section of this lecture is going to take me at least 5 minutes, so why don't we stop here at 3, and we'll pick this up on Wednesday at this point. I promise the punchline is coming. And please don't come to school tomorrow. You're not even allowed on campus. Any student on campus tomorrow will be shot and killed by the faculty. No, the campus is closed. Everything is closed. The library is closed. All the services are closed. There's no point in coming to campus. I will see you here on Wednesday. Study, study, study.